With innovative 3D pathology imaging and AI-enabled analysis in hand, GLORIA Biotech aims at solving challenging cancer diagnosis problems and fulfilling unmet medical needs for precision medicine. Deep imaging of each patient biopsy, followed by 3D image-inspired AI analysis, will bring customers value-added information of 3D tissue architecture with quantitative characterization, including cell number, distance, density, biomarker expression percentage and distribution profile, as well as relative ratio of regions of interest. GLORIA Biotech provides cancer landscaping report compatible with current pathology workflow. Software and AI support deep analysis of 3D pathology data, making affordability, efficiency, and sustainability possible in the new era of next-generation pathology.
Pathology examination is the key to cancer diagnosis, supporting clinical determination of disease stage and treatment plan. Hence, it is an important indicator for the cancer patient's life expectancy and decision-making. Traditional pathology diagnosis requires cutting a thin 4-micrometer slice from the patient's surgical biopsy, followed by morphology staining or biomarker labeling. Based on pathology features, pathologists diagnose cancer status with corresponding treatment guideline. However, heterogeneity of tumor tissues gives differential morphology features in different sections. A single slice contains too little information to represent the complexity of intact tumor tissues. In theory, pathologists can repeat biopsy sectioning many times to get complete information, but time and cost considerations make this impractical during clinical operations. If pathological examination relies only on a single slide image, it's hard to get a full picture of tumor features and precision diagnosis of cancer status, which makes the formulation of a suitable treatment plan and precision medicine extremely difficult in a clinical setting. GLORIA Biotech develops innovative 3D pathology diagnosis, solving the fundamental problems of sampling limitation and insufficient accuracy in clinical practice. GLORIA Biotech has integrated patented technologies to generate a comprehensive total solution for 3D digital pathology. Each biopsy collected from surgical operation will be processed and maintained in an ISO-certified laboratory with high-quality management of tissue clearance and tissue staining prior to retrieving reliable microscopic images for detailed analysis. GLORIA Biotech has developed innovative high-speed 3D imaging equipment to support the collection of more than 100 times high-resolution 3D tumor images and precision diagnosis that outperforms conventional pathology. We've developed the world's first specialized software, MetaLight, for 3D pathology diagnosis of morphology features. From big data of 3D tumor images, users can retrieve the most representative tumor tissue information as a basis for pathology diagnosis. GLORIA Biotech enhances diagnosis accuracy and lowers misdiagnosis with 3D pathology solution, benefiting cancer patients and providing affordable precision medicine. GLORIA, reimaging new horizons for pathology.
委员好，我们是丰康微流体晶片股份有限公司，那主要是做现场便利、快速的检测系统，那可以用一滴血10分钟就可以完成检测。
为什么你们认为在这个疫情过后，你们这样一个产品会是一个将来我们一般人生活上是一个很需要的东西？
精准医疗它就是其中有一部分就是希望能够提供更精准、更快速，然后马上就可以知道的一个检测结果。那我们也发现说，在这一次疫情，甚至在疫情之后，大家其实对于这个疾病的检测，甚至身体健康，还有传染病的预防，就会更为重视。所以呢，像我们是走这个 POCT 的检测系统，那我们可以在第一线、第一时间，不管是针对比较偏远的部分，或者是像这种必须要普遍筛选的时候，它可以在第一时间协助病患快速的检测。
Cell and Vision is a contract development and manufacturing organization, CDMO services company, for medical LDT lab tests and IVD medical devices. We are accelerating the discovery of identification and pathogenesis in reproductive medicine and oncology by revealing the true functional biology of every patient cell. Here, we provide fetal red blood cells isolation from maternal blood, which apply to fluorescent in situ hybridization, or real-time PCR, to aneuploidy analysis of chromosome 13, 18, 21, X and Y using cell-based liquid biopsy. Our products consist of immunochemistry fluorescence autoscanner and dynamic standing cell assembly cell array, DS-SACA chip. DS-SACA chip is usefully designed for rare cell isolation and standing without pumping system. As you can see, the target cell could be clearly isolated without pumping system in 30 minutes with 2 to 4 blood sample background ratio of a millionth. This automated scanning, microscope, and image analysis system is intended for in vitro diagnostic use as an aid to pathologists in detection, classification, and counting of cells of interest based on color, intensity, size, pattern, and shape of nucleated blood cells. There are some disadvantages for cell-free DNA-based NIPT. First, NIPT mainly detects fetal fragment DNA from ruptured RBC, but it does lose the ability to analyze complete genes. Moreover, maternal DNA interference, low signal-to-noise ratio need to rely on an algorithm to push back to improve accuracy. Our solution keep away those issues above and preserve all genome information of birth defects as early as possible. By looking for the fetal red blood cells in the maternal blood, it retains all the genetic information of the developing fetus, and its complete nucleus can be used to analyze the whole gene defect. Second, SACA chip provide highly specific screening of fetal red blood cells to avoid maternal cell interference. For the marketing analysis, the market of prenatal testing achieved $2 billion in 2019. As shown in the table, there are several antenatal examinations provided for pregnant women at advanced maternal age. Amniocentesis is usually considered as the gold standard in prenatal diagnosis. However, it carries a slight risk of miscarriage, and it should be specifically noted that more than 70% of pregnant women felt uncomfortable under the surgery. Instead of that, we're devoting to develop a safer methodology to reduce the risk of artificial abortion to meet the FDA regulation. So here, we provide a standard protocol by FISH or qPCR that are usually used in clinical practice, and is capable of taking the test at the earliest timing of the pregnancy period by our cell-based NIPT system, compared with the other detection technologies, such as villus sampling, NIPT, amniocentesis, 4D ultrasound. Side effects may occur with induced abortion, whether surgical or by pill. These include abdominal pain, cramping, nausea, vomiting, and diarrhea. Abortion also carries the risk of significant complications, such as bleeding, infection, and damage to organs. The occurrence of serious complications increased by more than 2 times with the lengthened pregnancy period. This SOP could help us get the high-purity fetal red blood cells in only 4 steps. The fetal red blood cells are separated from maternal blood sample by applying gradient centrifugation. By loading the separated cells onto the SACA chip, direct fluorescence staining or fish detecting can be conducted, which enables direct microscope observation on the chip. To further enhance the efficiency, we also developed a fluorescence autoscanner, which are established with integrated fluorescent imaging and analysis functions. And we validated the accuracy of fetal cell, which we capture with SACA chip by STR paternity testing. In this method, the fetal cell is set as experimental group and maternal cell as control group. The above example result shows the existence of Y chromosome from a baby fetal cell, which validates the cell selecting procedure. Moreover, we used the human prostate cancer cell lines to validate our FISH capability by successfully staining the target sex chromosome with commercialized FISH probe on chip. Using fetal cells in maternal blood can avoid the infringement of NIPT patent. Also, the current NIPT market acceptance is high, although there exist other similar platforms and products on the market. However, none of them have already been approved by FDA as IVD device. Cell Envision is working diligently aiming at becoming the first in the industry to gain FDA approval of cell-based NIPT IVD device.